Right. Thanks for that invitation. Thanks to ARI for inviting me again to talk about a topic that's very close to me, the one that I'm very passionate about. So I'm going to talk about 21st century developments in medicine, health sciences, and biotechnology. And I think the question is, why talk about this at OCON? And I think this is one area where it's not just the dismal world. So if you'd rather hear about the dismal world, there's lots of lectures on that. I'm not going to talk about a dismal world here. I won't talk about war, destruction, famine, authoritarianism. That's not in my lecture. And I don't know, maybe you'd want to focus on the November election. Maybe that's a better topic to talk about. So I'm not going to talk about that either. What I am going to talk about is the fact that humans are doing great things despite all of the death and destruction and horrible things that you read about in the newspaper or you turn on the evening news. You often don't hear about the things I'm going to talk about on the evening news because people don't seem to care anymore. They only want to hear bad news. And all these horrible things are happening, but people are doing great things. And they always have. And I think that's something that I want to try to concretize to you by sort of giving you an idea of this revolution that's happening in biotech that we're all a part of and seeing. I have this quote from the fountainhead from Rourke's speech, which I think is really emblematic of what the people in this field are doing. And it's just to read it: the creator is not concerned with disease, but with life. Yet the work of the creators has eliminated one form of disease after another in man's body and spirit, and brought more relief from suffering than any altruist could ever conceive. And I think that's what I'm going to try to concretize to you, how these things and what's going to build off of them are going to change the world for the better, despite everything else that's going on. So I don't know if any of you've all probably been to Epcot Center, and you've been in that geodesic dome, and they have the World of Tomorrow, that tour. That's sort of what I'm going to try to do in this talk. And I'm going to cover things almost like you're in a little tram car, different things that are going on in the health and sciences that I think are really important to take notice of. It was very hard for me to choose anything, so I picked about 6 things that I thought were really interesting to me, that I thought would be fascinating to this audience, but there's so many more things I could do, and each of these are a lecture in their own. So just bear with me as I go fast through some of them. So I'm going to talk about tools against mosquito-borne disease, CRISPR-based therapies, and as part of that, I'm going to explain what CRISPR actually is. AI-based antibiotic discovery, so using artificial intelligence to discover new antibiotics. Pig organ transplants, which I think is one of the most fascinating things, which should be the front page of the New York Times for multiple days, but it's not. Cell-based therapies for cancer, and some interesting stuff with new trauma resuscitation tools. One caveat, I'm a subject matter in medicine, infectious disease, epidemiology, microbiology, critical care, emergency medicine, not everything. So I'm going to be talking about things in molecular genetics and in hematology, for example, that I'm not a subject matter expert in. I think I know a lot about them by being a physician, but I'm not a full subject matter expert in all of those things. So if there is a hematologist in the crowd or somebody that I oversimplify something to just understand that part of it. And the other thing is I'm used to talking to medical audiences a lot when I give lectures. So if I use a word like progressive multifocal encephalopathy or something that somebody doesn't understand, I sometimes can rattle those things off. Just wave your hand that I've went over what you know about, because I have a tendency to just drop those words sometimes almost subconsciously. They automatically come out. So the first thing I'm going to talk about is the fight against mosquito-borne disease. And the last time I actually was in Anaheim, I was at the convention center giving a talk on Zika, which was something that I think got people very nervous about mosquito-borne disease. But I'm going to talk about how we deal with mosquito-borne diseases and what's happened in the last several years regarding mosquito-borne diseases first. So the most dangerous animal is not a lion, a tiger, or a bear, it's the mosquito. More than half of all people who have ever lived have died of malaria. So that's the biggest killer of humans has been malaria. Some people talk about it as the immune system of Africa, that's one of the reasons why Africa has not been able to develop is because of the mosquito-borne illnesses there. And I think it's important to think about mosquitoes not just as one species. There's multiple different species that spread different diseases. So we have Anopheles mosquitoes that spread malaria, Aedes, which kind of are the most prolific in terms of spreading diseases. Dengue, chikungunya, Zika, yellow fever, all spread by Aedes mosquitoes. And then Culex, which transmits West Nile. And this is just an ad from, this is Anne for Anopheles. This is something that Dr. Seuss made during the war because malaria is a major military issue, and that Dr. Seuss made cartoons about Anne drinking your blood. So what do? We tell people, avoid mosquito-prone areas, wear insect repellent, spray insecticides, drain swamps, remove standing water receptacles, insecticide bed nets. That's basically what we do. And all of these things have limitations. We still have about 1 million people dying every year from malaria and other mosquito-borne illnesses. And I think these are all good for what they are, but they don't really give you anything new that people didn't know about a long, long time ago. And then lots of people started thinking, instead of just trying to drain swamps and use insecticides, can we actually affect the mosquito populations permanently? So they started to move into these 21st century solutions by looking at how mosquitoes reproduce. So when you get a mosquito-borne illness, it's not the male that bites you and makes you sick, it's the female that bites you. And what that gave people an idea was, can you have somehow sterile males released that don't do anything, but when they mate with females, the offspring is not viable. And then does that decrease the population over time? Can you do that? And the initial way that they started to do it was irradiating mosquitoes, male mosquitoes, so that they're still going to be able to mate, but the males are infertile, and it doesn't produce viable offspring. So that was the first thing they started doing, is using sterile males, releasing sterile males that have been irradiated. And they took this from other agricultural examples, like, for example, the screwworm, which is a big plague on farms. But that didn't seem to be the best way to do it. That's more of a 20th century solution. The 21st century solution is to start genetically modifying the mosquitoes. So can you modify these mosquitoes to alter their reproductive systems so that they don't produce viable offspring by using gene editing? And they've been able to do that by releasing sterile males. And they call them friendly mosquitoes because there's so much antipathy towards GMO mosquitoes. And when you look at what they've done, I won't go into too much of the details there, but what you've seen is multiple countries have released these mosquitoes. So Brazil, the Cayman Islands, Panama, and India. And then one of the interesting things is actually parts of Florida. And a study I worked on was before Zika, they were thinking about releasing these mosquitoes. And I did my team at Hopkins, we put together surveys of this area in Key West to say, how welcoming were you going to be to GMO mosquitoes coming there? And you can imagine what the response was. It was no, nobody wanted them to come there. And it's been a very hard thing to get GMO mosquitoes used, even though when they're released, you can see 90% reductions in supply in the mosquito population. So they are very, very effective. That the mosquito population goes down, and this is something that controls disease outbreaks. And many countries that don't have the luxury of kind of avoiding mosquito-borne illnesses, like Brazil, like Panama, like India, have released them and have welcomed them. Even countries like Djibouti have released malaria with no controversy. But in the US, it has been very hard, but they have been able to be released in the Key West. And this is, I think, one of the elegant solutions to a scourge of humankind for so long that hasn't been amenable to insecticides, bed nets, draining swamps. But using technology like this, you really can gain an advantage. There's another way to do it as well. There's certain bacteria that you can inject into the eggs, which then cause the eggs not to be able to hatch in the individuals that actually bear those eggs. So this is a bacteria called Wolbachia. This is an easier way to do it because it avoids the GMO label, because it's not considered a genetically modified organism. So this is something that's been done in Fresno, California, for example, where they've released these types of mosquitoes out there to decrease populations. So these are two things that are already out there and that are working. These are all temporary solutions. So they bring down the population, but the population can move back in. The mosquitoes can regrow back up if you're not continuing to release them. One of the more aggressive solutions is something called a gene drive. So in genetics, when things are inherited, you kind of have an equal chance of inheriting or not inheriting a trait. A gene drive actually gives it a weight. So you can propagate a gene, like a gene to make you sterile, in a mosquito population. And that over time will cause that population to go down permanently. So that's the next kind of holy grail with genetic modification. It's not these temporary releases that bring things down, but to alter the populations of these target mosquitoes. And I think it's important just to go back. This is not getting rid of every mosquito. It's the mosquitoes that produce diseases like malaria, like dengue fever, like West Nile. So this is the holy grail, is to be able to do that. And it's very controversial because there's a lot of opposition to it, and people are worried if it can you reverse it? Are there reversal gene drives that you can use in case you need to bring back the mosquito population? I don't know many people who want to bring back a mosquito population, but these are the kind of considerations that are out there. And I would say, to his credit, does anybody know who one of the biggest advocates of gene drives are? Bill Gates. Yes, so I wanted to put a picture of him there. I like this picture of him. So Bill Gates doubles down on his wiping out of mosquitoes with gene editing. So he's been a major force here. And Bill Gates and the Bill and Melinda Gates Foundation has been instrumental in the control of some of these diseases. And I think that the genius of Bill Gates applied to global health infectious disease programs is just like the genius of Bill Gates applied to that Microsoft computer there. I wanted to put a picture of him with mosquitoes, but when you Google Bill Gates genetically modified mosquitoes, you go down a really weird rabbit hole of bad stuff that I don't recommend doing. So I found this picture of him when he was younger that I think is an interesting picture, but he's been a major advocate there. We do have vaccines against mosquito-borne illnesses, and I wanted to just highlight a couple that maybe people don't know about. I think everybody knows about the yellow fever vaccine, but we do have vaccines against dengue, as well as chikungunya, and then and Japanese encephalitis. But what's very exciting is that there are, for the first time ever now, malaria vaccines, 2 different malaria vaccines that are being used in Africa. These decrease morbidity, mortality from malaria, 70 to 80%. That's really, really important, because if you think about Africa, you think that this malaria is a poverty trap for those countries. They cannot economically develop very well when so much of their resources are spent controlling malaria. So I think that this vaccine is going to pay dividends, and you're going to see, I think, industrialization, people being able to survive through childhood, not have these malaria episodes. All of that can really be instrumental for bringing civilization to some of those countries that have been kind of lagging for some time. So lots of good stuff going on with mosquito-borne illnesses. So now I'm going to move to CRISPR and understanding that. So everybody's heard of CRISPR, right? Does anybody know what it stands for? The molecular biologist can't answer if there's any. I think I saw a couple walk in, but yeah, there she is laughing, yeah. So CRISPR stands, most people who say CRISPR don't know what it stands for. Clustered regularly interspersed palindromic repeats. Does anybody know what any of those were? So basically what it is, it's a certain genetic sequence that repeats itself that's in genes in certain areas. And where does it come from? Where did CRISPR come from? What is its natural role? It's basically a very primitive bacterial immune system. So I think mammals and vertebrates have these really, really extensive immune systems that can respond to lots of different things. Bacteria most people don't even really think they had an immune system. Or why do bacteria even need an immune system? Do bacteria get sick? The answer is yes. There are viruses that infect bacteria. And why bacteria involved this CRISPR system was to basically take copies of the genetic elements of the viruses that infect them and then store them in their own genome, basically keep a copy of that. So when they see this bacteria again, they can cut up its genetic sequence and make it non-viable. So CRISPR is a very primitive immune system that bacteria use to fight off the viruses that infect them. And I think that's really astonishing that occurred, but you can just see that if you've got a system that can copy a genetic code and then find that code and just cut it up, that's basically really, really elegant scissors. And that's what happened, is people realized that this system could be harnessed to do editing of genes, not just to fight viruses that infect bacteria. So 2 people that won the Nobel Prize, Jennifer Doudna and Emmanuel Charpentier, they're the ones who discovered the ability of the CRISPR system to be used for gene editing. Now, this is where it's going to start to get a little bit technical. So what happens when you get gene edited, or a human gets gene edited? Basically, what they do is they take your cells, and they take these cells out of you. They do this fancy CRISPR stuff. I'm going to show it to you in a second. And then they put them back into you. In the meantime, you get chemotherapy so that to get rid of all the bad stuff that's there, the genetic. So you have a genetic disease. They kill your bone marrow, take the cells out, edit them, and put them back in. That's basically what gene therapy is. And I just want to emphasize that this part is not easy. That says myeloablative conditioning. Conditioning is not like running on a treadmill. That means destroying your bone marrow. So this is, you're super immunosuppressed at this period of time until these cells engraft. And that's kind of where, as an infectious disease doctor, I play here when they don't have an immune system. But that's what happens. So it's still not like you get a gene, an injection, and you're better. You still have to do all of this in the hospital, which is pretty cumbersome. So what does CRISPR do? So scientists create a genetic sequence called a guide RNA that matches the piece of DNA they want to modify. So you've got some gene that you want to cut out. They get a guide RNA, they stick it on this other protein, Cas9, which guides the whole system there, and it basically snips it out and either can put in a new piece of DNA or just cuts it out. So that makes it's a very major oversimplification, but they're precision-guided scissors that can go after a gene of interest and cut them using that same bacterial immune system. So it's this guide RNA that's really important. The guide sequence is coupled to the cutting site, and they can go in there and cut it. So we have gene therapies using CRISPR for diseases related to hemoglobin. So I'm going to take a detour just to explain to you these hemoglobin diseases so that you actually can understand how these work. So a couple of things. Everybody's heard of the word hemoglobin, right? Yeah, so hemoglobin is the oxygen-carrying molecule on your red blood cells. That's what, when you breathe in air, breathe in oxygen, it binds to the hemoglobin in your red blood cells in your lungs, and then that red blood cell with the hemoglobin takes it to your tissues and releases the oxygen. So it's a very, very, very important molecule. And it has a really particular structure to it, which I'm going to get to in a second. And there's different forms of hemoglobin. When you're an embryo, you have a certain type of hemoglobin. When you're a fetus, you have a different type of hemoglobin. And when you're an adult, you have a different type of hemoglobin. And that has to do with the structure. So I'm not going to spend too much time on this. It's not super important to memorize any of this, but just so you have this hemoglobin. It has the adult hemoglobin has 2 alpha chains and 2 beta chains. That's the structure. Fetal hemoglobin has the alpha chains but has gamma chains. And at 6 months, your body switches from this fetal hemoglobin with the gamma chains to those with the beta chains. And after 6 months, you only have about less than 1% fetal hemoglobin. It's all adult hemoglobin. And there's a special enzyme or a protein that's responsible for that switch. But what's important is if you have a problem with a beta chain, then you don't have the hemoglobin is not going to be structurally normal. It's not going to work. So there's certain diseases, which I'm going to get to in a second, where you don't want the adult hemoglobin, you want fetal hemoglobin. But this switch here has stopped that from happening. So what happens is this switch basically takes you from adult hemoglobin to takes you from fetal hemoglobin to adult hemoglobin. And the question is, can you edit out that switch, just cut it out, so that you don't switch and you continue to make fetal hemoglobin? And that's really important for 2 diseases. One all of you have heard of, sickle cell anemia. So sickle cell anemia is a mutation in the beta chain, and what it causes is abnormally shaped red blood cells that form this sickle shape and then can not deliver oxygen very well, get stuck in your blood vessels, and cause horrible, horrible disease. The average lifespan of someone with sickle cell disease is about 20 years. So it's a horrible disease. Does anybody know why genetically sickle cells stayed out? Why didn't everybody with sickle cell die? Yeah, malaria. So going back to mosquito-borne illnesses, from an evolutionary perspective, this has been selected by malaria. So people who are carriers of sickle cell, they don't get malaria, they don't get sickle cell, but if a carrier and another carrier have a child, that child could have sickle cell. So that's why it's persistent. So what we've done is try to induce hemoglobin F. There's certain drugs that can cause hemoglobin F, the fetal hemoglobin, to go up. But that doesn't really work very well, so that's why they wanted to move towards using CRISPR to just stop that switch, increase fetal hemoglobin so that these people don't sickle. And the same thing is true with another disease called beta thalassemia, which probably not many people have heard of, but that means not enough beta chains due to mutations, deletions, and people there become anemic, dependent on transfusions. Because of the transfusions, they get overloaded with iron. So these two diseases are very kind of low-hanging fruit to show, can CRISPR work? It's interesting from a philosophical thalassemia, that it's Mediterranean water, Thales, that's how I remember it. People in the Mediterranean area is where thalassemia is more common, from a history of philosophy tidbit there. So there is a drug, some of you maybe have heard of it, called Casgevy. That's what it does. It basically decreases, it removes that gene so you don't get that switch to adult hemoglobin, or that you can make more fetal hemoglobin. And you can see in the clinical trial in the New England Journal of Medicine, their fetal hemoglobin levels went up. So these were path-breaking discoveries for these blood diseases that had no cure, nothing that you could actually do for these individuals except for a bone marrow transplant. And this is now the first application of CRISPR to human disease, which is extremely fast, because CRISPR was just in the headlines as being discovered. People talked about how it could be used, and now it's already being used with FDA-approved products for two diseases, which I think is really just the beginning. And some people might talk about the price of these, that they're very expensive. For example, Casgevy's probably, I think it's a couple $1 million for the infusion, but it's still cheaper than what it takes to take care of a sickle cell patient. So they're very cost effective, even though they've got a high sticker price. But I think this is an incredible story, that this technology has developed so fast within a decade or so. Has everybody got this? Moving on from CRISPR to something else. Is there any question? Everybody got the CRISPR stuff? So the next thing I'm going to talk about is antibiotics and better ways to discover antibiotics. So we're in this situation with antibiotic resistance where there's a slogan, bad bugs and no drugs, that it's becoming harder and harder to treat certain infections because we just don't have antibiotics that work. And there's few to no classes of new antibiotics discovered. When you hear about a new antibiotic being approved by the FDA, it's usually a me too or a version of something that's very similar to what's already existing. It's not something path-breaking. It's not something new. We're running out of antibiotics. And one of the things that constrains this is that pharmaceutical companies aren't very interested in antibiotics. The economic model is not really conducive to investments in antibiotics. If you think about an antibiotic, you take it for a week. If you get a very fancy new antibiotic, the first thing people like me, infectious disease doctors, are going to tell people, don't waste it, don't use it, we need to save it, we need to keep it on a shelf, or use it only with our approval. And what does that do? It gives them a very, very, very low return on investment. They want a drug like Prozac or Viagra or Lipitor, something that people are going to take all the time, and there's really no bound to it. But an antibiotic, it's like precious gold to us, and that is not conducive to people wanting to invest in antibiotics. So we've seen more and more companies that are in the antibiotic realm go bankrupt, more and more people not want to even invest in antibiotics. They move on to other pharmaceuticals because it's just not the greatest in terms of return on investment. And the idea of antibiotic resistance isn't something new, even though we're hearing about it now, that there's headlines, there's a big UN meeting on it going on right now. This is something that Alexander Fleming, who was one of the discoverers of penicillin, said, that it's not too difficult to make microbes resistant to penicillin in the laboratory by exposing them to concentrations not sufficient to kill them, and the same thing has occasionally happened in the body. So he's writing that in the 1940s. This is something that's now come kind of full circle, where you see all of these scary headlines all the time that we're in this issue where there's no new drugs. And what that threatens to do is pull us back to the pre-penicillin era, where it becomes very hard to do anything. You think about antibiotics, they're used for not just treating infections, they make all of medicine possible. So from putting in a hip replacement, to putting in a cardiac pacemaker, to doing transplant surgery, to doing chemotherapy, to doing routine surgery, all of that requires antibiotics to prevent infections. So this is why, in my field, this is considered one of the biggest threats is antibiotic resistance. And it's very hard to get companies engaged in this. It takes about $1 billion or $1 to $2 billion to bring a drug to market. And so many of them fail because they don't work or they have toxicity problems. So one of the things that we're hearing about now is using AI or artificial intelligence to do drug screening, to predict what a molecule might do. So how is AI used to facilitate the development of a new antibiotic? So property prediction model. So properties possessed based on the structural characteristics. So they can look at all these molecules. They're basically shifting things. Remember, antibiotics were coming from the dirt most of the times. They come from, for example, fungus that will release substances to kill their competitors, or bacteria that release substances to kill their competitors. That's where most antibiotics come from. But you've got to sift through all kinds of molecules to figure out, does this work, does this not work? But what AI can do with machine learning is understand what properties are possessed by these molecules. Are they likely to have effect on a bacteria? And then that can kind of narrow the search and make it much more efficient for a pharmaceutical company than to narrow in on these compounds. It generates a set of molecules that can then have further testing. So this makes it much, much more efficient to look for antimicrobial activity or antibiotic activity in molecules. And then you can even use generative AI to create molecules from the building blocks with specific properties. You can ask an AI to make an antibiotic or make something that has a property to kill this bacteria by using this mechanism, and that can actually occur. So what we're seeing is a major step forward in the way that we think about antibiotics that makes it easier, less cost prohibitive, and faster, which hopefully will get us to a point where we're able to have new antibiotics, because this is one, as I said, the most pressing challenge in our field is this problem. I just have some slides here some pictures that are not projecting very well, but basically what that shows is how they're predicting what a molecule, what properties it may have if it has effect against a bacterial species. So this is just a way to speed it all up, but I think it's very exciting because we've had really no innovation in the antibiotic space, and this is one way that AI, you think about AI a lot with diagnostics, helping to interpret chest x-rays or mammograms, but it even has this application here as well, which I think is going to be a lot of what we see in AI is going to be, there's the diagnostics and electronic medical records and making a doctor's life easier, but it's also going to be making the other side of this easier of like being able to pile through lots of data and find patterns that can then be useful for somebody to do something in the future with. So next stop on the tour is a very cool one, and this is organ transplantation and xenotransplantation. So what's the problem we're trying to solve with organ transplantation? There's 93,000 people on the waiting list for kidney transplant in the United States, 10,000 on the waiting list for liver transplant, and about 4,000 in the heart transplant list. There are not enough organs to transplant. I mean, there's lots of reasons for that. There's lots of inefficiencies. We don't compensate donors. We don't compensate donor families. All of that makes it harder to get these lists down. And I don't see anybody wanting to do that to compensate donors. So what people have been trying to do, and they've been trying to do this even before they started to transplant human organs, is xenotransplantation. I have a picture there of Icarus because some people say that Icarus is an example of transplantation because the guy transplanted wings onto a human from another, from a bird species or whatever it might be. But that's a picture of Icarus there because some of the xenotransplantations use him as an inspiration. So initially, people tried to do xenotransplantation by using blood products, by using skin, by using corneas, trying to get them to work from one person to another. And then in 20th century, people started seeing even rejuvenation therapies with baboon and chimpanzee testicular transplants to rejuvenate males didn't work. And that became kind of the paradigm. People tried to put baboon hearts into humans, things like that. It wasn't quite working. But what's happened is that we've gotten to a point where this shortage has become dire. People are dying on the transplant list all the time, and people are looking, is there a way to use an animal organ in humans? Based on all that stuff we talked about, CRISPR and gene editing, can you make a human organ, a pig organ or whatever organ compatible with a human? And lots of people kind of centered on pigs because they're about the same size. The organs are about the same size as humans. And they are easy to raise. They have a short time to maturity. And we use lots of pig products in medicine already. Sometimes somebody might have an artificial pig heart valve. We've had pig insulin, for example. So porcine products are used a lot already in medicine. So there's not so much of a taboo to using them. and these are the right size and easy to rear. The challenge is, though, with organ transplantation is rejection. Humans reject other humans' organs. That's why we do all this stuff. You see these things for looking for a match for a kidney or a match for a bone marrow, or whatever it might be. They need to be very exquisitely matched to the donor, or your body is going to reject. And how do we stop it from rejecting? We give people immunosuppression. And what problem does that cause? It makes them very susceptible to infection. So these are the problems. And they think if you're giving something from a pig, you're going to have to use lots of immunosuppression. And you're worried about not only the ordinary infections that the person might get being in the hospital or being out among people, but can you get an infection from a pig with an exotic virus that's only in pigs? How do you stop that from happening? So these were big challenges. So what did they do? With the challenge of rejection, how do they meet that challenge? Basically gene editing, using all of those tools we talked about. They basically take all of these basically what they call is depigging the pig, basically is how to put it. They take 4 pig genes related to cell surface antigens and growth hormones and just delete them, and then they insert 6 human genes. And you don't need to know what these genes are, but there's 10 genes, so it's a 10 gene edited pig. And that becomes basically humanized. And then they raise these pigs, and then they harvest the organs from those pigs. So there are colonies of these pigs that exist that are humanized, that are less likely to reject. And I think this was a major advance that couldn't be done before without better gene editing tools that we now have. The other thing is infection. The pigs have endogenous viruses in their genetics, and you don't need to get into the details of it, but this is something that is something they had to think about. Can you rear these pigs without certain endogenous viruses? Some of them you can't. It's in all pigs. There's some that aren't, so they're doing that kind of selection because they don't know what these pig viruses will do in humans. So the first thing they did is they didn't just put this into any human. They first started on brain-dead individuals. So this was done at places like NYU, places like in Alabama are the big pioneers here. So what they would do is someone is brain dead, they get consent from the patient's family, they take out their kidneys, and then they put the pig kidneys in, for example, and see, do these pig kidneys make urine? That's what kidneys do. They filter your blood and make urine. Does the person reject? And they started to have success in these individuals that were brain dead, which was easier to do from an ethical standpoint because you didn't know what would happen, and from getting it through the IRB at a hospital and getting FDA approval to do these things. So they did that. That was successful. And then they quickly moved to humans. So these are some of the major breakthrough papers. So they did hearts and kidneys first. And the first heart patient was at the University of Maryland. He lived about 2 months. He was somebody that could not get a regular heart transplant, wasn't a candidate for anything. He consented to it as kind of a pioneer, and he lived about 2 months after that pig heart was put in place before he developed an infection with one of these pig viruses that flared up and likely caused his death. Then the second one was done again at the University of Maryland. This person lived about 6 weeks and then eventually developed rejection of the organ and died. But that man got to go home from the hospital, and he would have been dead much earlier than 6 weeks were it not for that pig organ. Then they gave kidneys to living patients as well. The first kidney transplant person died 2 months afterwards, unrelated. They say they've not really released any details. They said it's unrelated to his transplant. That's all we know. And kidney 2, basically she got a kidney, but they had to take the kidney out because of surgical problems that happened with it. But these are the first people that have had these organs, which are kind of the first pioneers that this is possible. And these are people who otherwise would have been dead much probably quicker than had they got those organs. And I think they're getting better at perfecting this technique and understanding it. In China, they did put a liver in a human, and we don't know much about what's going on in China, but they said doing very well. So there are advances going on all over. And I think it's really important to think about these as the first kind of forays into something that's probably going to change transplantation forever. Eventually, we probably will be growing organs. It's very hard to do that now because people don't quite understand how to make an organ as functional as the one you need to put in you if you grow it in the lab. you can't, for example, an embryo has to be destroyed at 14 days. You're not allowed, I think, by federal law to keep an embryo going, so they can't actually even study embryogenesis that very well in a test tube to see how well you can do this afterward, how well you can make a heart, what does the heart look like when it's growing on a petri dish in a human embryo. So there's some restrictions on that. So I think this is going to be the next thing that happens, and then eventually we'll get to this 3D printing of organs maybe decades from now, depending upon how the environment is to work with embryos, because you have to. I was going to skip that slide. So these are two companies, just if you're interested in this, Revivicor and eGenesis. They're the ones that are pioneering pig transplant organs, and I have no financial interest in any of those companies. They're just really cool companies that are doing a lot of stuff that's at the cutting edge. And one of the guys, I think at eGenesis, is the same guy that's cloning the woolly mammoth. And if you follow biology, he kind of looks like Darwin. His name is George Church. He's a very visionary biologist, but he's part of this group that's been doing that. So really cool things that are going on there. Next thing, cell-based therapies for cancer. Have people heard of that concept before, cell-based therapies? Yeah, so what does that mean? So when you get cancer, part of your immune system doesn't just attack bacteria, viruses, fungi, parasites, protozoa. It also does something, tumor surveillance. It's always looking around and usually wiping out cancerous cells. And people thought, can you boost that immune surveillance, that tumor surveillance that's going on? And can you engineer cells to attack cancer cells? And people had thought about vaccines, doing things like cancer vaccines. That's all coming around. But one of the things that's already out there is something called CAR-T. Has anybody heard of that concept? Yeah, CAR-T. So CAR-T stands for chimeric antigen receptor T cell. So that's one of those words that you should wave your hands that you don't understand. But basically what it is engineering T cells. So in your immune system, you've got B cells, T cells. T cells are something that can be engineered to go and attack tumor cells if you put the right receptor on them. So if that T cell can recognize this is a cancerous cell, I'm going to go and kill it. That's basically what they do. So what they do in certain cancers, primarily blood cancers, and I'll tell you which ones in a second. In blood cancers, they take the T cells out, they modify them in a laboratory, and then they reinfuse them. And when those things go in, they're like revved up, ready to go go and kill tumor cells. So they basically go there and wipe out the tumor cells. And this has been kind of become the treatment of choice for many different blood cancers. So certain types of lymphomas, leukemias, multiple myelomas. So the FDA has approved it for multiple different types of cancers, and they've become really successful. When you infuse those cells, though, they do cause a lot of side effects because your body's in a war, because those immune cells are really going after the cancer cells, and they go into what we call like a storm type of atmosphere in that patient, and that can be challenging to manage. But these really have revolutionized the way we treat these leukemias, lymphomas, multiple myeloma, and I think that it's only going to be, over time, this is going to be something where you don't have to do this step, that you don't have to make them in the lab, that they're right off the shelf, that you can just have them. So now there's a process where the person has to have their T cells harvested and sent to a lab, and then given. They're trying to eliminate that step and have what are called off-the-shelf CAR-T therapies, and they may start using CAR-T therapies for solid tumors. So these are all blood cancers, leukemias, lymphomas, myeloma, all of those are cancers of blood cells. The thing next would be to try to do solid tumors like pancreatic cancer, kidney cancer, those types of things. That's where the next step is with cell-based therapies for cancer. But I think it's a very exciting aspect of the field. Another one that I think is really cool, and this is from one of my home institutions at the University of Pittsburgh, and that's one of the professors that I actually trained under there in this picture, Michael Pinsky. But basically, what they've done here, at the University of Pittsburgh, there's a thing called the Center for Military Medicine. And the military is very interested in treating patients or people that get shot in the field and getting them fixed before they get to a forward operating hospital, because there's lots of people are going to die right away. So what Pitt has done is made this device, and they just tried it on an animal. They basically stab an animal, and you hook it up. You put the lines and the IVs in to this animal, and the computer decides how much blood this person needs, how much of the IV fluids it needs, all of the stuff. It feeds back off of the animal and autonomously decides how to resuscitate. And what they did in Pittsburgh is they call it precision resuscitation. They put a pig in hemorrhagic shock and then stuck it on a helicopter and just flew around Pittsburgh to see, can we save this pig, as if it were a real person, like you're coming to a car accident or a gunshot wound, and you just plug them into this machine, you put the IVs, and that's the only thing the humans had to do, were put the IV catheters in, then the computer did the whole thing. And they were able to successfully do it. So I think this is going to be really, really interesting for military applications and for trauma applications, where you have basically a robot machine that decides, this person needs this much blood, the blood pressure is doing this, I'm going to do this, this is going up. It's going to really make things much easier, because lots of people die in that first hour after a traumatic accident. They call it the golden hour of trauma. And if you can do something like this, you may be able to save a lot more people, because that transport time, all of that is a time where mortality goes up considerably. So this is something that is really really cool type of stuff, and it's really cool that it's happening in Pittsburgh. So things I left out, just a couple things. Suspended animation, has anybody heard of that? That's another trauma thing. That's where someone gets shot and you cool them down and you just suspend them, you fix what's going on and then warm them back up. That's going on at the University of Maryland. That's also a military trial for trauma that's looking promising. Bacteriophages, I talked about those viruses that infect bacteria. They can be harnessed to kill bacteria that infect us. So people are using bacteriophages to treat drug-resistant bacterial infections. So you find the virus that kills this bacteria and you give it to people who are infected with that bacteria. There's been major advances in that field as well. eCPR, so that's a type of CPR that's not just pushing with your chest, it's pushing people on the chest and putting them on a ventilator. It's hooking them up to a heart-lung machine where they basically get almost what I just talked about, but they get it allows them to get blood flow until you repair what's wrong with them. So people who go into cardiac arrest, they do CPR, they don't come back, they'll declare them dead. These people get hooked up to a machine and they can be sort of revived, and you fix what's wrong with them in that period of time, and they may go on to live. So that's just a whole major change that's going on with CPR. And then the other thing that I wanted to say is we've got hepatitis C cures right now. We also have long-acting HIV drugs, which have revolutionized the treatment of HIV. So lots of interesting stuff going on. And we also have our second mRNA vaccine that was just approved by the FDA last week by Moderna for RSV. We have other RSV vaccines, but this is the second mRNA vaccine that's on the market. So we're seeing the mRNA vaccine kind of renaissance start to happen already. So that's all I had, but I left, I have 17 minutes for questions because I knew there'd be lots of things that people would want to ask about that I talked about, or things that I didn't include, and I'm happy to take questions on all of that. I hope that was interesting. I hope that made you think that the whole world isn't dismal, that there are people doing really cool things, and hopefully that inspired some of you to look these things up.
Thanks for a great talk.
Well, okay. What's new about it's just a new mRNA vaccine. The RSV vaccine is a new. We already have 2 RSV vaccines. This is the 3rd one, but this is the one using mRNA. But it's the 2nd ever mRNA vaccine. We have one for COVID and now for RSV. Yeah.
What are some of the regulatory barriers that you'd like to see knocked down, and is anybody working effectively toward that?
So the regulatory barriers are when you move to human trials and making sure that the FDA is okay with all of that. And I think for most of these major breakthrough innovations, the FDA has been very permissive with kind of discretionary enforcement and working with the companies to make it happen. And you have to kind of take the FDA's perspective on most of their tasks, that they don't see themselves as barriers to this. They think this is our role to do this. How do we work with these companies to make this pass regulatory muster? So oftentimes the FDA is working with these companies from the very beginning and trying to make it happen. So in my mind, most of these things haven't been constrained so much by FDA regulations. It obviously adds costs to be able to do when the FDA asks for another study or not, but I think the FDA's goal is to get these things through, and they have certain designations, like they call this a breakthrough therapy, so that means that they'll kind of keep their hands off of it. So it's more about making sure that the companies have clear you're not going to have wholesale, the FDA is going to be in place for a long, long time. The key thing is making sure that the FDA has clear guidance and works with the companies so that they're not kind of blindsided by things and then a whole research program goes down the drain. For example, with the embryos, I definitely think there needs to be some change there on having the embryos be allowed to be worked on after 14 days, but that's not an FDA issue, that's a religious, political issue, so it's not, so, in most of these things, I don't think the FDA has been that constraining in this field.
Hi, thank you for the great talk. My question is on antibiotics. Do you think AI can help create new antibiotics more frequently so we don't need any prescription for antibiotics anymore? Or is this a political issue and not a technological issue?
Well, so I don't think you'll have a point where the AI just makes you. I mean, that would be really cool, like in the Jetsons, that cartoon where you just go to your fridge and you push a button and an antibiotic comes out. But I think that there's probably with antibiotics, I think you're going to want to have a medical professional's advice on using it to make sure it's the right antibiotic for the infection that you have. What I think AI will do is make it easier to create antibiotics for pharmaceutical companies, develop new antibiotics, rather than remove the need to consult a doctor. You can imagine, though, that we have with telemedicine and all of this, all these advances in technology, that it would be very minimal, that you can think about, you have a home diagnostic test at home, like we have home COVID tests. Imagine you had a home test for strep throat. You test yourself and it says, okay, your thing is positive for strep throat, and it automatically will give you that. That, I think, could probably happen for certain conditions. But I think for complicated infections, you're probably still going to need a medical professional to help you figure out what antibiotic you need.
Thank you.
I found the example of the fetal hemoglobin's really interesting. Once the patient has fetal hemoglobins, are there downsides to that? Obviously, it's preferable to sickle cells, but
not really. No.
Oh, that's interesting.
Yeah,
Why does it switch then?
Because the fetal it has to do with the way oxygen is transferred through the placenta, and the fetal hemoglobin. You need your mother's hemoglobin to release the. If it was the same, the oxygen. That's going to get technical. So the oxygen has to move across the placenta from the red blood cell of the mother to the red blood cell of the fetus. So if the hemoglobins have the same binding to oxygen, it's never going to release.
It'll never
so they
switch.
have different. So the fetal hemoglobin binds oxygen more readily, so it will pull it off of the mothers.
Do they get better? Do they get like better cardio after switching to fetal then?
I don't know if they do. I don't think we've studied that to see if they do. But yeah, I don't know if they would.
Hi, Amish. I just had to take the opportunity to have an expert tell me directly. Can you comment on the safety and efficacy of mRNAs? I think your last slide talked about it, but just say it to my face.
Oh, so there's no specific safety concern with mRNA vaccines. I think that they have been game-changing when it comes to COVID-19. All vaccines have certain side effects, but there's none that I think that are really untoward with the mRNA vaccines that are not, we've seen, with COVID vaccines, there has been some increase in myocarditis, but very mild myocarditis. But no major thing that I've seen that's a safety signal. It's important to know that mRNA, we have it in your body anyway. mRNA is something that's been around, that they've been working on for decades. And the safety signal is no different than any other type of vaccine. And I think they really are a step forward in vaccine development.
Thank you so much.
I don't know that there's a answer to my question that's not context specific, but my cousin in India, she got multiple myeloma, and 3 years ago she got the CAR-T treatment, went to Spain, spent a lot of money. It was interesting because back then it seemed like this was almost a foolproof, they were talking about how 10-year patients who had this 10 years later were still in remission, and that it was almost like a guarantee of success. And then a year later, it actually ended up that she got her cancer back much stronger, and ended up getting allogeneic transplant, and is actually now, seems to have gotten past. But curious about what some of those things might be, which would take something like CAR-T and why it would not work, and maybe there isn't a way to answer that in a generic way.
Well, so the reason why it might not work is it might not kill all the tumor cells. There may be some that resist, or some that it doesn't get, and then those are going to be much more aggressive and harder to kill. But yes, CAR-T doesn't kill everything. They don't get it all. I mean, you can get relapses. I think it's extremely successful in a lot of patients, but there are going to be individuals where it doesn't work completely, and they might need another treatment or a different type of treatment, like a standard bone marrow transplant. But I would defer to an oncologist if there's one in the crowd.
This is all wonderful and exciting, and wondering what you might think about what's next, like say in the next 10, 20 years. What do you see, asking for a prediction, but I'm curious to know what you think.
So I would say more mRNA vaccines, not just for infectious diseases, but for cancer. Better diagnostics, more precision-guided treatments that are based on an individual's genetics or the actual risk that they themselves have, rather than kind of one-size-fits-all treatments. I think you'll probably see pig organ transplants become much more common. I think that they'll probably work and they'll get it perfected. I think you'll have more gene editing therapies, not just for sickle cell and beta thalassemia, which are very low-hanging fruit. I think they'll see more complex. Better treatments for Alzheimer's disease, better treatment. I think, what else is going on? Muscular dystrophy type stuff. All of those types of gene therapies are going to flourish, I think. Hopefully in your home you'll have more diagnostic tests to be able to test yourself for lots of different things, more telemedicine going on, hopefully less mosquitoes, hopefully those malaria vaccines will work, and we'll see more of that, and maybe see a West Nile vaccine eventually as well. I guess it's hard to predict. Every day I had to keep updating these slides, like every day there's something new, and I think that's a good thing, but I don't know where it will go. It's hard to predict exactly where people's minds will go, but I think there's a lot of convergence going on in biotech, with bio and with tech, that are converging using tools like AI to make things work a lot faster and easier. So I think it's really, and then I didn't mention the Neuralink stuff that Elon Musk's company, that's also something that's very, very good. And maybe even, I know you work for Theranos, maybe a real Theranos will appear.
Thank you.
Hi, thank you very much for your talk. I wanted to just clarify something. Are you talking about, from the first subject, significant reduction in the planet's mosquito population at some point?
Yes.
As someone who, I would be the last person to defend mosquitoes. I think when I moved out of the Midwest, the mosquito population dropped by half from starvation. But is there any drawback biologically or ecologically to not having mosquitoes?
Not particularly those mosquitoes, because most of them are, it's not, remember, this isn't every mosquito. We don't want the bats to starve and
right, I mean,
and birds eat them. So no, these are very targeted to specific mosquitoes that spread disease. And for many of them, they're invasive mosquito species that didn't exist in those places for a lot. They came, so for example, like the Asian tiger mosquito in the United States is not native to the United States. It came and migrated here. So, and it's not all mosquitoes. Some of the good ones will still be around to bite you.
Thank you.
I'm about as much as a layman as humanly possible on subjects regarding medicine, but I was reading into some stuff for some biotech investment opportunities, and one of the things that frequently came up, I think, since COVID and recently has been technology that allows for the transportation of certain mRNA is partially revolutionary, I think, because of the ease of transportation of the drug rather than some other things that are kind of hard to get throughout the body. Do you see anything related to, I guess, transportation of drugs within the future, or that's already happening?
So talking about drugs being able to get to different body tissues. So
yeah.
With mRNA, what actually made it able to do that was the lipid nanoparticle that it's encased in, rather than the mRNA itself. But yeah, there are trying to use nanotechnology to get drug to the actual site. So that there are major advances going on there as well. I'm not as familiar with them, other than the ones that are being used for mRNA vaccines to get them to sites. But they are trying to get drug delivery exactly where there's even something called like radioactive drugs that can go directly to a specific, it can home in on certain cells. So that is happening. I'm less familiar with it though.
Okay, thank you.
I really enjoyed kind of learning an optimistic viewpoint on genetically modified animals, because the only other time I think I ever get exposed to those ideas is in Jurassic Park movies. We obviously culturally have a fear of this kind of stuff, and I'm curious, do you have any insights on where you think this comes from, and is any of it based on any kind of reality? Have we ever created a genetic monster we've regretted, or is this entirely in the realm of science fiction?
It's entirely the realm of science fiction, but I mean, there's a big philosophical underpinning to it. It all goes back even to Mary Shelley Frankenstein, where that's basically talking about kind of the hubris of science, where Dr. Victor Frankenstein wants to create life, and he creates this monster, and that's supposed to be this tale that you shouldn't be meddling with these types of things. So this goes back probably, to probably the Dark Ages, where people were not wanting people to do anything, use anesthesia, for example, on patients, or to dissect human bodies, where they had to steal them from graves. There's been kind of an anti-science attitude for a very, very long time, and GMO is just one aspect of it. And as I said, Frankenstein, Godzilla, all of those things are anti-science in terms of this is what happens when humans meddle with certain things, that you get monsters like that. And with GMOs, it's become kind of on the right and on the left, where they've this is tampering with nature. In Florida, for example, lab-grown meat is banned. You can't do that there. And then on the left, you've got all the granola crunchy people that don't want anything, that they don't want it. So I think this is something that's widespread, that you're tampering with nature, even though Native Americans tamper with nature all the time, because the corn they found is not the corn that they were eating. They modified it by crossing it and making it more nutritious by doing it the old-fashioned way, not using CRISPR, but by doing it by mating different types of corn. So this is something humans have been doing for a long time, but people have been opposed to it for a long time. And I think it's really, it definitely makes it hard for companies, because if they get labeled GMO and people set fire to Monsanto fields, and if you say you like GMO, then they think you're part of this whole conspiracy that's going to poison everybody with golden rice and all these things. That's rice that has vitamins in it, actually. But there's just this, it's completely backwards, and I think it's hampered the entire field. I have stickers that say GMO that I keep on my fridge. I just like them. There's a company called Ginkgo Bioworks, which I do hold shares in, but they're really pioneering genetically modified everything, and they give you these little stickers that say pro GMO type stickers, which I think it's important to do. That I want things to be genetically modified. I'd rather have it genetically modified and not have like normal E. coli in it, like a Chipotle burrito, where there's not. I pick on Chipotle because they pride themselves on that, and then they had major outbreaks because of the way they prepare their food, and I'd rather have it genetically modified and irradiated and all of that than have the normal E. coli swimming along in there. But yes, it's a long-standing anti-science type of thing that goes back for a long time.
We have a question from the online audience. Are there any advances pending in the area of complex differential diagnoses, especially distance services?
So differential diagnosis is a term of art that we use in medicine, where we're trying to figure out what's making my patient sick. And yes, there are work with AI models to be able to take a patient's symptoms and give likely diagnostic pathways for those people. And some individuals with really complex problems have used ChatGPT-4 and have then taken that output to their doctors and said, can I be tested for this? And they found the diagnosis. So yes, there is work to use AI for complex diagnostics, even not just interpretation of something, but typing in symptoms and then in your test results, and then it figures out what it might be. Yes.
What would have to change in the economics of the vaccine development in order to make creation of new vaccines profitable?
I think. First of all, vaccines would have to be embraced. That's one thing. That the companies don't want to get involved because that's a litigious area to begin with. Number two is that they get seen as an essential need, and that pushes governments and organizations to want to cap profits, not want to pay a lot for them. So companies don't even want to get engaged in it because they know they're not going to get a big return, because many of the diseases that we want vaccines for affect people in the developing world, where they don't have purchasing power to be able to pay enough to get a proper return on investment. So a company doesn't want, if they make a malaria vaccine, they're going to be pushed to sell it at cost. And that doesn't make sense for investors. So I think that there is a lot of altruism that's embedded in the global health world, where people don't think that these companies should make a profit, or that they should have intellectual property. For example, they'll say, well, then license the intellectual property so this can be made here for no cost, but you can make your profits up in the first world or in economically high countries or high income countries. but I think it's just not something that they get a good return on investment. When you can think about how much they make, for example, on cancer chemotherapy that's sold only in the US and Canada and the UK or in Western Europe, that's where they're making their profits. Vaccines just are not that, they just never have been that profitable, and it's something you give them once. And so a vaccine you give to people, it's not the same thing as making a drug like Lipitor where someone's going to be on it for their life. So the return on investment just never made that great of sense. And then all of the other factors have made it harder and harder for companies to justify that to their investors. mRNA is, I think even when you look at Moderna, which is the big mRNA pioneer, their major business proposition was vaccines for cancer, not vaccines for infectious disease. That was kind of a side project that they were able to capitalize on, but it's not really what they were thinking that they were going to claim their fame to. So I think that there's just, the infectious disease realm is not very profitable for most pharmaceutical companies, not just vaccines, but also antibiotics, antivirals. They're just not, they just haven't been as lucrative as cancer therapies or lifestyle medicines.